by these tribals. In India, this culture is practiced mainly in the northern eastern states and Assam. It is also found in Bihar, West Bengal and Orissa. When coming to the Muga variety, this golden yellow color silk is prerogative of India and the pride of Assam state. It is obtained from which is obtained from multivolatile silkworm Antheraria assamensis. These silkworms feed on the aromatic leaves. Aromatic means the one which produce the aroma that smells better is som and solu. These plants are available in Assam. Muga culture is specific to the state of Assam and an integral part of tradition and culture of that state. The Muga silk and high value produced silk is used in products like saris and mekhalas and chaddas. This is very highly valued silk. When coming to the life cycle of silkworm, here as we already discussed in the last week, this is the life cycle of the silkworm. You can see here the normal egg, that is diapause egg. Eggs are hatched and the first instar will happen in 4 days. That is first molt stage. Then second instar, you can see here 2 to 3 days, second molt. And third instar, 3 to 4 days, third molt. And fourth instar, 3 to 4 days, fourth molt. And fifth instar, that is 6 to 8 days. The matured and ripened silkworm you can see here. And the way of cocooning, the messy and the mesh type silk is producing here you can see. Then the fresh cocoon with a live pupa harvesting which is happened in between 7 to 8 days. Here you can see the fresh cocoon. Then once the fresh cocoon in the time of 3 to 4 days, it will become like a butterfly. uh, Here this is the silkworm life cycle. A silkworm cycle on Bombyx mori, a silkworm life cycle on silkworm Bombyx mori. Here you can see the same thing which just we discussed. The silkworm feeding on mulberry leaves. These are the mulberry leaves, and how the raw silk is producing here you can see with the colored pictures. And the hatching days 3 to 4 days. When coming to the egg laying, and you can see here from the mating, egg laying, egg, the developmental stages inside the egg into 10 to 12 days, then hatching 3 to 4 days, and first mold stage just now we discussed 2.5 to 3 days, and the second mold stage 3 to 4 days, and the third mold stage 4 days, and the fourth stage, that is fourth and final mold, 6 to 8 days. When the silkworm is ripe, the spinning silk filaments you can see here, it looks like a network like structure. And this is cocoon with a complete spinned silk, and a cocoon shell. When we cut open, here you can see the pupa stage and emergence from the cocoon, 10 to 12 days, and again mating on the mulberry, that is rasil. With this introduction, our next topic is mulberry cultivation and farm maintenance. How can we cultivate the mulberry and how can we uh, maintain the mulberry farm? Here I'll show you the instruments which we need and how we have to maintain the mulberry farm in what are the important steps we have to take for avoiding the pest and the pest control methods also we will see. As we know, sericulture is an important art of production of cocoons and silk. What are the mechanization of mulberry farming? Here, what are the mechanics and what are the plantation methods to cultivate the mulberry plants? What are the tools, equipments, and machines for the cultivation of the cultivation of the mulberry? Here we will see. As we know, you understood. Just we discussed about the sericulture and what are the importance of the sericulture. Sericulture is an art of production of cocoons and silk. You understood that it consists of production of mulberry leaves and rearing of silkworms. Though sericulture has been practiced in many parts of our country for centuries, the total production is much less than the national requirements. Today, the Indian sericulture industry is facing a shift challenge from foreign silk, mainly from China. Just now we discussed about the Chinese silk because Chinese silk is superior in quality and also cheaper. Hence, you must make a real effort to improve quality and reduced cost of the production of indigenous silk. The Indian sericulture industry is highly laborious and intensive and expensive. Over 60 to 65 to 70% cost of cocoon production is accounted for by wages for different activities. Under these circumstances, mechanization is considered as a critical input for reducing labor and involvement and bringing down the cost of production. Before going to this, here Before going to this, here we have the types of silkworms just we discussed. Those are mulberry silkworms, tussar silkworms, oak tussar silkworms, muga silkworms, airy silkworms, gonometa silkworms, fa silkworms, pagara silkworms, cone silkworms, Indian moon moth, mussel silkworm, and spider silkworm. How these are distributed and the different hatching stages and how many days it will take for the production of a good silk, everything we studied. So here the when coming to the summary, the four species like just we discussed, the tussar silk and temperate silkworm and oak silk, airy silk, all these are very important silks. When coming to the taxonomy of the mulberry, mulberry trees are perennial, live for a number of years either in cultivated or wild conditions. Depending on the type of cultivation, the plant is grown as a bush. Just now we discussed about the silkworm. So now we have to discuss about the mulberry trees. Why? Because anyways, our silkworm is depending upon the mulberry. So that we have to understand the importance of the taxonomy of mulberry trees and morphology and anatomy of the mulberry, and also we have to know the systematic position of the mulberry. So as I said, this is a perennial plant. It lives for many years either in cultivated or wild conditions. Depending upon the type of the cultivation, the plant is grown as a bush, tree or a middling. The branching nature of a plant is once again influenced by type of cultivation and mode of training. It mainly depending upon the soil fertility, rainfall and environmental conditions. However, profuse branching is must for producing more amount of leaf as so to feed silkworms. The motto of this plant is feed for silkworms. Since the cost of production of cocoons reflects the efficiency of leaf production, utilization of leaf by silkworms. When coming to the taxonomy of the mulberry plant is mainly to produce high quality and high quantity of the leaf for rearing silkworms. The leaf quality has positive effect on the quality of cocoons, which directly influence the silk yarn quality. The silkworm Bombyx mori being monophagous and host specificity is due to the presence of attractants in the mulberry leaves, but not mainly due to the nutritional value. Mulberry leaf protein is the source for the silkworm to biosynthesize the silk, which is made up of by two proteins. One is fibrin and sericin. These two are the proteins which is presented in the mulberry leaf, which gives the source for the silkworm. But mulberry includes a number of species and varieties. We'll see what are the and varieties in upcoming. They differ in their suitability for silkworm rearing because of their varying nutritious value and palatability for the worm. The Central Silk Board has recommended certain specific mulberry varieties to suit the eco-climatic conditions of a particular area. In order to have an ideal about these entire aspect, one should know that about the mulberry plant morphology and anatomy. What is the systematic position of the mulberry? As we know, this mulberry plant belongs to the Morus, which includes 35 species. There are more than 1,000 species of plants belonging to the family Moraceae. It belongs to the family Moraceae. It includes wild and cultivated forms, some of which are sterile. The method of classification of mulberry species should therefore based on genome constituents or on external characters like leaves, flowers, fruits. Lediber, he is the scientist in the year of 1846 to 1851. He classified Morus alba and Morus nigra on the basis of papillae and pubescence on the stigma. Bureau in 1873 used external characters such as length and shape of female inflorescence and syncar and divided the Morus alba into two. Brandis classified the mulberry species into two sessions by the length of the style, and Koizumi he grouped mulberry into two sessions on the length of the style of the female flower. Hotta, he divided the mulberry species into two groups on the shape and position of the cystolith cell in leaves. When coming to the taxonomy, it belongs to the kingdom, plant kingdom, and division, phaneogamy, subdivision, angiosperms, class, dicotyledons, subclass, monoclamidiae, series, unisexuales, family, Moraceae, genus, Morus, and species, Alba. Based upon the species, there will be different types of species, that is Morus indica and Lavigata serrata. In India, we have different kinds of species. Those are Morus alba, Morus indica, Morus lavigata, Morus serrata are widely grown in Himalayas. The classification of Indian varieties is also much confused based on principles of the classification adopted by the Hota. Guru Rajan, in the year of 1960, he suggested grouping all the cultivated varieties of mulberry into three species. One is Morus alba, that is from Mysore, Tamil Nadu, and Andhra Pradesh, and Morus bombycis, Berhampur, Morus latifolia, that is presented in Kashmir and UP. Here you can see the structure of Morus bombycis. Yes, this is the structure of Morus bombycis and Morus alba and Morus latifolia. These are the mulberry varieties. And Hooker, he is the scientist who described Hooker, he is the scientist who described Morus. He is the scientist who described the Morus in detail. The Moraceae family is identified by the presence of idioblast. There are long cells in the upper epidermis of the leaves which contain cystoliths of non-crystalline life. What is the habitat? Habitat means where it will grow. It, it grows as a tree or shrub as we discussed, but in cultivation it is raised as bush by pruning. Pruning means cutting. The plant is perennial with highly branching roots and shoot system with primary, secondary and tertiary branches. The branching character varies considerably due to the influence of different factors such as a type of cultivation, mode of training, fertility of the soil, and mainly rainfall, means the humidity of the soil. However, the mulberry cultivation is mainly aimed on the branching to produce more leaves, which is suitable for the silkworm rearing. The trees attain a height of 22 to 25 meters with a width of the trunk about 8 meters in case of Morus serrata. In multivoltina areas of India, Karnataka, West Bengal and Andhra Pradesh and Tamil Nadu, mulberry plants are grown as bushes. The univoltine areas like Jammu and Kashmir, it is grown as a tree. Mostly it looks like a bushes. When we go for the pruning, it will show the bush-like character. The mulberry bush attains a height of 1.5 to 1.8 meters, which would be an ideal height for leaf harvesting. When coming to the Stem is a aerial portion of the plant which supports the foliage of the mulberry. The stem is divided into nodes and internodal regions, and the color of the bark of the stem varies from green, gray, and gray to pink, and also brown with numer of lenticels. Like it looks like a fiber, looks like a hairy region. Most of the branches are erect, means straight, but sometimes they are trailing. In nature, mulberry grows a big tree reaching a height of 20 to 25 meters with a width of 8 meters. These characteristics are varies depending upon the species. We have different types of species, and the characteristics like height and width varies depending upon the species, and also it depends on the climatic conditions and origins. As we discussed, the length of the plant mostly depending upon the rainfall and humid conditions of the soil. The Mysore local variety Kanwa 2 and Berhampur varieties of Morus indica are white to grayish white in color. In temperate regions means that the area which has more temperatures. The leaves fall off in autumn and the bud comes in the winter, so called as winter bud. The winter bud has 7 to 8 scales on the outside and about 10 young leaves inside. Sometimes between the scales and young leaves within the winter bud, the smaller birds looks like a scaly birds. Means in between the scales and the young leaves, there will be a presence of winter bird. That are smaller birds called scaly birds. The mulberry of tropical climatic sprouts throughout the year. The buds are of two types. One is vegetative and other one is reproductive. Coming to vegetative buds. Vegetative bud gives rise to vegetative parts like leaves and branches. Reproductive birds produce male and female inflorescence in addition to leaves. The rate of sprouting of vegetative birds is of primary consideration in introducing a variety of species in an area. Thus, these birds are important prerequisiting in the spring sprouting of plant in temperate regions. And in vegetative propagation of plants in both tropical and temperate regions, from the cultivation point of view, the birds which are on the shoots less than 1 year are called as normal birds. When shoots are trimmed, when shoots are cutted and trimmed, all normal birds existing on the remaining parts of the shoots sprout and grow. But the birds at the bottom of the shoots become latent birds. Usually in spring season, some birds at the top and bottom of the shoots do not sprout. Unsprouting birds at the middle part of the shoot play an important role at the time of effecting countermeasurement against the frost damage. The birds which do not grow due to extreme climatic conditions means high temperatures or low temperature, high rainfall or low rainfall means drought regions. So those are called as extreme climatic conditions. In that time, it will go to the resting period called as resting birds. Winter resting birds are found in mulberry. Mostly it happens in the winter. In winter, the resting birds are found in mulberry, so called as dormant period. The dormancy in birds may be attributed to lower temperature and nitrogen deficiency and inactivation of enzymes due to excessive accumulation of carbohydrates. Photoperiodism means presence of excessive amount of H2O2, and the presence of auxins. Like there are recent research happened. These recent findings indicated that auxins are responsible for dormancy in bird. This chemical is more in terminal bird, can be broken by mechanical chilling or chemical treatment. Here, as just we discussed, there are winter resting birds found in mulberry. Those are called as dormant birds, which is happened, which is mainly attributed to because of low temperatures and nitrogen deficiency, mostly attributed to inactivation of enzymes due to excessive accumulation of carbohydrates, sugars, and photoperiodism. And yes, there is a studies findings indicated that the presence of auxins are responsible for dormancy in bird. This chemical has more in terminal bird, can be broken by mechanical chilling or chemical treatment. When coming to the bird, each axil of the leaf, the axil means the part in between the stem and leaf branch. Each axil of the leaf has a bud. Sometimes two or more independent birds we can see. Main birds are found are called as accessory birds. The axillary birds are green in color that turns brown latter stage. The axillary buds of the scale leaves of the main bird peep out of the bird as though they are the accessory birds. Each bird is covered by 2 to 3 layers of the scaly leaves of brown in color with hard texture. These protect the birds from frost and high temperature. These are called scaly birds, scale birds. The birds is protected by the covering of young leaves which are in turn covered by the older and oldest leaves. Below the scaly leaves, the foliage leaves, that means older leaves, are found which overlapping each other, and the apical meristem is situated in the center. There are two types of birds. One is vegetative bird and reproductive bird. Vegetative birds gives rise to male or female inflorescence in addition to leaves. The growing points emerging as a shoot under favorable climatic conditions. Here you can see, here you can see the sprouting bud. Here you can see the sprouting bud and axillary bud, and the foliage leaves just we discussed, and how the scaly leaves looks like, and this is the bud sprouting axillary buds and Morus indica axillary buds. There. When coming to the leaf, leaf is the most economic unit, as we are discussing from first um, term. We are talking about sericulture industry, mainly depending upon the Morus indica, that is mulberry leaves. So as per that knowledge, we can consider the leaf as a most economic unit in the sericulture. which serves as the main food for silkworms. It should be the aim to here our aim is to increase the leaf yield as per the unit area. Leaves must be succulent, smooth, less fibers and palatable to the silkworm that have to give the harbor to the silkworm. The leaf size of the mulberry varies in different species and varieties. The leaves are simple and alternate, stipulated and petiolate and entire or vigorously lobed. The leaves are alternate and ranked with different internodal lengths. Lobation starts from unlobed to highly dissected type. Like lobation, the leaf apex, base and margin also differ widely. The stipule protects the young leaf. As the leaf matures, the stipule drops off. The stipule size varies with variety. The petiole length varies from 2.5 to 7.5 centimeters with a groove on the dorsal side, means the back side of the leaf. The leaf may be glossy, looks like a glossy and scrapers in structure. If you will touch the leaf, looks like very glossy. The leaf may be entire or lobed, but rarely both types are found in the same twig. In general, the hybrid leaves are unlobed while local varieties are mostly lobed. The number of lobes per leaf varies from 1 to 7 and in Sujanpur the leaves are 5. The leaf is highly dissected with numerous lobes. The leaf lamina is usually glabrous but may vary with the variety. The size of lamina nearly measures 30 centimeters in length along the midrib and about 25 centimeters breadth at the widest region. The thickness of the leaf is about 100 to 200 meters. The shape of the leaf is generally ovate cardate, looks like a heart shape and also looks like a egg shape. The base of the leaf may be cordate, means heart shape, or straight and truncate or ordinary cuneate. The leaf tip is generally of four types: acute, double acute, acuminate, and long acuminate. The leaf margin may be serrate, dentate, dentate serrate, serrate dentate. Means it looks like a arrangement of the teeth. The venation of mulberry leaf is multi-costed reticulate venation. The leaf base is generally three-nerved. The phyllotaxy varies according to species, it depending upon the species. It is alternate or spiral. Indian varieties are five ranked. The surface of the leaf, if epidermal cells is covered with cuticle, is a protective layer that protects the leaf. There are narrow lines on the cuticle and these lines have a relation to lustre of leaf. In the upper epidermis of mulberry leaves, there are large cells called as idioblasts. The idioblasts contain cystolates in which non-crystalline line that contains the calcium carbonate. Calcium carbonate. That's what it looks like a crystalline. If you can see that particles under the microscope, you can see the needle-like structures. The hybrids leaves such as Canva 2 and S54, S36, S30, S34 that have been characterized by large leaves while Mysore variety local beer only have small leaves. And coming to the inflorescence, in mulberry, it is a catechin. In mulberry, more than 10 flowers grow around the rachis, being attached to the axil region of new shoots, and such type of inflorescence is called as spike or catkin. Catkin is with pendant of drooping peduncle bearing unisexual flowers. The plants are generally dioecious, but the occurrence of monoecious plants is also not uncommon. Rarely, bisexual flowers are seen due to development of pistillats, staminodes. The male catkin is usually longer than female catkin, measuring 2.5 to 5 centimeters long. The inflorescence is axillary and branched in certain cases. Usually, they are in clusters. The female catechin is axillary in position, measuring 1.5 to 3.5 centimeters long. When coming to the flower, the male flowers are loosely attached to the peduncle. The flower consists of four perianth leaves arranged in two whorls with imbricate estivation. That is, the anti-posterior perianth leaves are outside, while the leaves in the transverse plane are inside. These are green in color, homochlamydious in nature. The margins and outer surface of the perianth leaves are sometimes provided with hair. The stamens are four that arranged to perianth leaves. The filaments of the stamens are bent inward in bud condition when it is in bud condition. Each stamens has a broad filament and a narrow top on which two anther lobes are fixed. Anthers are longitudinal dehiscence it shows. The pollen grains are round, that is dry, light and dust-like with a smooth exine. When the bird opens, the stamens which lie in curved position become straightened. Suddenly, the andrecium opens with the strong force and the pollens are scattered from the anther. This is an adaptation to the wind pollination. After pollination, male catkin becomes pink in color and dries and it droops off as a whole. The female flower also got similar number of perianth leaves arranged in same manner as in male flower. These leaves persist in the fruits. The fertilization stimulates the female flower and perianth leaves which then become thick, containing juicy material and contribute to form the fruit. The ovary is superior and bicapillary syncarpous, oval in shape, unilocular with a single pendulous ovule attached to the margin of the ovary. The pistil consists of a style, a stigma, and an ovary. The pistil originates from the ovary and has a bifid stigma. The length of the style is botanically significant character in identifying the various species of the mulberry. Based upon the style, we can classify the mulberry species. The style are long in delicostyle and short in macromorus. After pollination and the fertilization, the entire inflorescence become a multiple fruit. When coming to the fruit and seed, cross pollinate when the result of the cross pollination in fertilization latter, the style and stigma fall down and the ovarian wall swells and changes fleshy making fruit. It is called syncarp or sorosis fruit. After fruit formation, inner part of the ovarian wall becomes endocarp which shows lignification and covers seed. The syncarp is in green color at first and later becomes red and dark purple. The seed in dark purple colored sorosis gets the ability to germinate effectively. The fruits are ovoid to subglobular in shape, measuring 5 centimeters in length. It is oval in shape with a nearly flat surface at the micropylar region. On one edge, an elongated streak hilum is presented. The end of the hilum has a small pore known as micropyle, through which seed absorbs water when soaked. The seed coat has two layers. One is outer hard and brittle testa, and the inner thin looks like a papery and slightly brown in color called tegmen. The seed kernel consists endosperm and embryo which lies curved in the endosperm. The embryo has a primary axis, contains plumule and radicle and two cotyledons. During germination, the plumule gives the shoot and radicle forms the root. The diploid species seeds weigh less than polyploid seeds. The mulberry seeds are variable for longer period at low temperatures, that is 5 degrees centigrade, or low relatively humidity and than high humidity. When coming to the root, before going to the root, the mulberry root has a tap root, lateral roots and root hair. The hair is confined to the top of the root. The root surface has many lenticels. The root hair absorbs water and nutrients from soil, and the lenticels perform respiration of roots. The lateral and tap root transport and preserve water and nutrients. In general, depending upon the soil conditions, uh, uh, since before we are discussing about the plant structure, everything, always we are uh, discussing about the soil condition. If the soil conditions are good, then the plant will grow nicely. So the soil conditions, the roots penetrate to the deeper layers and depth ranging from 10 to 50 centimeters. This is about the plant description. When coming to the anatomy of the root you can see primary root is a typical of dicotyledonous plant. The epidermis presented as a single layer which contains the tubular shaped structure. Cells, those are closely arranged with inner cellular spaces. The cortex is next layer of the cells. The cortex which is made up of by the parenchyma. And there are a clear single layered epidermis which gives the protection to the root, and coming to the endodermis which gives the protection to the pericycle and vascular bundles because it enclosing the pericycle and vascular bundles. What are vascular bundles? Here the vascular bundles are diarch in nature without cambium. The pericycle is many layered, which latter form the cork cambium. Here Morus indica root shows the secondary growth. In roots starts from the formation of the cambium from some of the parenchymal cells outside the protoxylem. Then cambium strips divided and redivided to produce the sinus cambium layer, which latter becomes the circular cells. This cambium cuts cells both outside and inside, and simultaneously some of the pericycle converts into meristematic cells to form cork cambium, which latter produces produces secondary phloem. The primary xylem, which remains undistributed as main medullary ray below the cork, is the secondary phloem. The secondary phloem xylem are formed from the activity of the cambium. When coming to the stem, the primary stem consists of the single layered epidermis with the tubular cells without intercellular space. The outer cells of the epidermal layer are cutinized. Cutinized means presence of cutin that protects the stem. Some of these cells are drawn into elongated unicellular hair, which protects the stem from the injuries from the insects and also minimize the rate of transpiration. Transpiration means evaporation of water from the stem. Below the epidermis, there is a multi-layered cortex presented which consisting of parenchymal cells. Some of the cells contain chloroplast. There are many lactiferous cells in the cortex which store the organic excretory products. The single layered endodermis with the conspicuous barrel-shaped cells and pericycle is multi-layered. The vascular bundles are very few, collateral and conjoined, open and endark. Endark means inside. There is conspicuous medulla presented. The secondary stem consists of vascular bundles with the union of cambial stripes. And a continuous ring of cambium is developed which cuts off secondary xylem towards inside the secondary phloem towards outside. As a result of increase in girth of the stem, the epidermis is broken at various places. Below this, a layer of cork cambium, phylogem, and inside this, a layer of secondary cortex or phylloderm. The secondary cortex consists of parenchyma cells transversed by the lactiferous cells. The secondary phloem consisting of the well-developed sieve tubes to consume food material and conduct the food material. Due to increase in the girth of stem, the primary xylem is pushed to the center, which ultimately crushed to form hardwood. The amount of wood produced in the 1 year in a annual rain. When coming to the leaf, we already discussed about the leaf. Yes, leaves has the dorsoventral and reticulate venation. The taxonomy of the leaf, upper epidermis consisting of the single layer of the tubular cells without any intercellular spaces. It contains the idioblast and those are the storing parts of calcareous secretions that contains CaCO2, calcium carbonate, and the outer cells are cutinized. Below the epidermis, there is a layer of palisade tissue with elongated cells containing chloroplast. The next layer is spongy parenchyma with the intercellular spaces. The lower epidermis is interrupted by a large number of stomata. Each stomata is guarded by two kidney-shaped guard cells and supporting subsidiary cells. Vascular bundles are found parallel and the midrib portion has maximum vascular tissue. Vascular bundles at this portion are arranged in omega shape. When coming to the petiole, the internal part of the petiole, its structure is like primary tissue of the stem. The epidermis presented in petiole is single layered Some of the cells are drawn into the epidermal hair in certain species. Below epidermis, there is a supporting tissue of 4 to 5 layers of parenchyma. The ground parenchyma is like the cortex of the stem. The vascular bundles are collateral and scattered in the ground parenchyma in a circular manner in the early stages. The phloem is oriented towards the periphery of petiole. Here you can see the structure of here you can see the structure of transverse section of mulberry leaf and a portion enlarged. Just now we discussed about idioblast, palisade parenchyma, cystoliths. Cystolith means calcium carbonate presented here, and this is the cuticle, which is a protective layer. Upper epidermis, spongy parenchyma, lower epidermis, epidermal hair, and this is the xylem and phloem and parenchyma, collenchyma. And when coming to the leaf, as we discussed, it contains the cuticle is a protective layer. Upper epidermis is also a protective layer. And the cystolith, here you can see the cystolith and parasite parenchyma, spongy parenchyma, and respiratory cavity means presence of stomata and lower epidermis. Here, mulberry plant is the main to produce high quality and quantity of leaf for rearing silkworms. When coming to this summary, the leaf quality has positive effect on the quality of the cocoons, which directly influence the silk yarn quality. This plant belongs to the genus Morus, which includes 35 species. There are more than 1,000 species of plants belonging to the Moraceae. In India, there are many kinds of species of which Morus alba, Morus indica, Morus lavigata and Morus serrata are widely grown in Himalayan regions. We discussed about this, and it grows as a tree, shrub, and when pruning, it looks like a bush. Here, I have a question for you. The Morus species present in the how India are. What are the Morus species in India? Yes, here we have Morus alba, Morus indica, Morus lavigata and Morus serrata that grows in the Himalayan regions of India. The plant is perennial with highly branched roots and shoot system with primary, secondary and tertiary branches. So the other one is what is the like mulberry stem is divided into how the mulberry stem is divided? Yes, here roots are shoot system is divided into primary, secondary and tertiary branches. And the mulberry root system, yes, it contains the highly branching root system. It is an aerial position, the stem is the aerial position of the plant which supports the foliage of the mulberry. The stem is divided into nodal region and internodal region. Means nodes and internodes are presented in the stem. Each axil of a leaf has a bud, sometimes two or more independent buds on side of the main buds are found are called as accessory buds. It is most economic unit in the sericulture, which serves as the main food for silkworms. It should be aimed to increase the leaf yield per unit area. Leaves must be succulent, smooth, less fiber, and palatable. Palatable means eatable to the silkworms. The shape of the leaf is generally ovate cordate. The base of the leaf may be cordate and straight and truncate or ordinary or cuneate. When coming to the inflorescence, mulberry is a catkin and has the 10 flowers grow around the rachis, being attached to the axillary region of new shoots. Such type of inflorescence is called as spike or catkin. The main flowers are loosely attached to the peduncle, the flower consisting of the four perianth leaves arranged in two whorls with imbricate eastivation. When coming to the flower, that is female flower, also got a similar number of perianth leaves arranged in same manner as in male flower. Fruit and seeds show cross pollination that results in the fertilization. Latter style and stigma detached from the tree, the ovarian walls swells and changes fleshy, making fruit. The mulberry root has taproot, lateral roots, and a root hair. When coming to the anatomy of primary root, is a typical one and dicotyledonous plant. The epidermis is of a single layered tubular shaped structure closely arranged without intercellular space. The primary stem consisting of single layered epidermis with tubular cells without intercellular spaces. The outer cells of the epidermal cells are cutinized and mulberry leaves are dorsiventral with reticulate venation. The upper epidermis consisting of single layer of tubular cells without any intercellular spaces. And the petiole structure is like primary tissues of the stems. Here we can see the questions just now we discussed. The Morus species present in India are Morus alba, Morus indica, Morus langivata and Morus serrata. And the other question I asked you is mulberry stem is divided into how? Nodal region and internodal region. That is nodes and internodes. And the other question is the mulberry root. How the mulberry root is presented. It is a taproot which contains the lateral roots and long hair. Canva and the other one is name some mulberry hybrids. What are the mulberry hybrids here? Canva 2, S54, S36, S30, S34. This is about this is the complete description about the taxonomy of mulberry plant. Sir, shall I leave? So this is about today's topic.